So welcome to Unit 3, Review. And in this unit, we really kind of broke it up into 2 or 3 parts. But we're just going to review as one large topic about cellular energetics. So what we've covered in our class was this idea of metabolism, which is made up of anabolic and catabolic pathways. And we focused on this idea that energy within the cell is using ATP, or adenosine triphosphate. We also talked about the idea of feedback loops as a way to maintain and control our metabolism. And then we broke the energetics into 3 major topics. The first was enzymes, the second is photosynthesis, and the third was respiration, specifically cellular respiration. Now with the enzymes, we focused on the idea that there are proteins, and they are function about proteins can affect enzymes because they are proteins. So we looked at reaction rates and factors that may affect the structure and therefore the activity. And so we focused on pH or temperature or substrate concentration. We also looked at cofactors, inhibitors, and feedback loops within the enzyme systems to talk about how they're controlled. And every arrow that we see in these chemical reactions is an enzyme. Now, photosynthesis and cellular respiration, we focused on the parts, in this case, the organelles. We focused on what reactions and what controls those reaction rates, the biochemistry. And then within photosynthesis, we compared plants that are called C3, and we looked at C4 plants and how they fix that carbon dioxide. And in cellular respiration, we looked at 2 pathways, aerobic and anaerobic. So when I go over these slides, what we covered, this is where I would suggest that you would come back, maybe go through your notes, go through your book, and see if you can kind of find the highlights, the important parts for that information. But today I want to do is focus on just one animal and how it fits into this puzzle. So we're going to focus on polar bears. So what I would like you to do is you're going to pause the video here. And as you're pausing the video, would you kind of look up, how much do you really know about a polar bear? Do they hibernate? Do all of the bears hibernate of the polar bears? Maybe only some do? I don't know. What is their main food source for polar bears? And how many subspecies are there? So I'm going to go ahead and let you pause here and go find that information. Welcome back. So did you find out some of those answers? Because they're going to need it for this next set of slides. So we've been talking about metabolism, so let's take a look at a research paper I found about enzymes and metabolic pathways comparing the polar bears versus grizzly bears. So take a minute if you want. You can pause it and read the abstract. If you remember, the abstract's the summary of the research. So you can go ahead and read through that, but I will give you the highlights. They looked at the mitochondrial DNA and they looked at the nuclear DNA between polar bears and bears, and they were looking to see if the genes between the animals are different, and was the difference caused by the environment or by just plain genetic drift. And in some cases, they did find some mutations that were unique to the polar bears that may have an effect on specifically cytochrome C oxidase and another pathway called nitric oxides. So it's pretty clear that the enzymes for the polar bears are slightly different because the animal has to survive in very cold, frigid temperatures. And in some cases, the animals literally have to stand still or lie on the ice and just generate heat through metabolism just to keep warm. So that's going to require enzymes that can function at a very low temperature. And it turns out the fur isn't the best at insulation, and the blubber is really where that protects them, but it's also the energy source to keep them warm. So let's kind of take just a quick overview of enzymes. So we know that the enzyme acts on something, it's called a substrate. They bind together in an enzyme substrate complex. And we've talked about this idea of an induced fit or a locking key method on this active site. And so really this induced fit is how the protein can change shape as the substrate binds to the active site. And so it's very, very specific, yet it still has to move, it still has to change. So here's a computer model that shows how it changes. That's called the induced fit. Let's do that one more time. So you see how the protein can bend and change so that it kind of forces the substrate into that active site. And this idea that mutations can change a DNA and then affect a protein, then maybe those mutations may affect enzymes and maybe make it into something novel, something new. So in this example I'm going to share with you, there were 6 amino acid changes that occurred near the substrate binding in the E. coli bacteria. And these changes allowed for this enzyme to work better in E. coli bacteria. So the mutations, sometimes we consider them bad if they're lethal, but may actually have some effect on improving proteins. And maybe these mutations within the polar bears allowed for the polar bears to survive in those very cold Arctic conditions. So let's focus on those pathways. So remember, each arrow of a pathway is an enzyme, and each enzyme has a substrate and a product. And then those products can then be used to either bind to a site directly on the protein or an allosteric site, or actually act as an inhibitor to play a part in a feedback system. And the one you're looking at now is a negative feedback system. So as product is formed, it then turns and feedbacks onto the protein, changing its shape, blocking the active site, and preventing the enzyme pathway from continuing again. So we've talked about pathways. We've talked about the idea that there's anabolic and catabolic pathways. So what I want to do is focus on catabolic pathways known as aerobic respiration. And this is really how bears are getting that energy and this food to maintain their body temperature in these cold, frigid environments. So here's a general pathway, or in this case, a catabolic pathway showing you carbohydrates, fats, and proteins. And as they enter into the different parts of the glucose to the mitochondrial pathway to oxidation phosphorylation. So you'll notice that at different points you can enter any one of our biomolecules into the pathway. You'll notice where it says proteins, you'll see where it says NH3, that's the removal of the amine group. That would then enter into the circulatory system to be filtered out in the kidneys, and then that ammonia would become part of the molecule called urea and eventually urine. So let's kind of focus in on this central pathway, glycolysis, citric acid cycle, and the oxidative phosphorylation. Of course, this all deals with the idea of the mitochondria and aerobic respiration. And we focused here, if you remember, on something called oxidation phosphorylation. And we talked about the process of oxidizing one molecule and then using that energy to reduce another molecule. So you have glycolysis, and you see these electron acceptors called NADH and FAD. These electron acceptors, these molecules, would accept the electrons at different energy states and bring them into the mitochondria, where they would be sent down the oxidative phosphorylation electron transport chain. There you would generate far more ATP than you would at something called a substrate level phosphorylation, which is what you would see only in glucose. So do not spend your time trying to memorize all the biochemistry here. The important part here is to understand that there are processes that take place in the cytoplasm, glycolysis, There's a process that takes place on the inside of the mitochondria. That's your citric acid cycle. And there's a process of generating ATP in the membrane level. So this is aerobic respiration. Here we compare that idea with anaerobic, or something called fermentation. Now in fermentation, this process is just using glycolysis and then creating lactate in animals or ethanol from the plant sugars and the organisms that live on plants. Again, the idea here is to understand that the pathway can go in two different paths, and so with or without oxygen. Okay, so we know that the base of the food chain are the producers. So in this slide, I found a research about modeling the Arctic sea ice. So we understand that there are certain pressures right now in terms of climate change, the ability for the ocean water temperatures to increase, and the melting of the sea caps. That could there be a change in the algae blooms, and when they happen, and if so, how would that affect the food chain and eventually the food web? And more specifically, what would happen to the animals, in this case the polar bear or the ring seal or the fish, if these algae blooms changed because of changing climate? So if you want to pause it here and read this abstract. Welcome back. All right, here's our overview. Again, I don't want you to spend a lot of time on the biochemistry. That's not the important part for the test. Here I want you to understand what's happening though. So there are two components to plant photosynthesis. There's a set of reactions that require light and water, and those are the light reactions. They require two systems, series of chemical reactions called photosystem 1 and photosystem 2. Here we do an oxidative phosphorylation in reverse, where we're then splitting water to release the oxygen to grab an energy from the electron. Then that electron is then passed on to an electron acceptor. We also have a substrate level phosphorylation, and then that is sent into the stroma, or the space around the thylakoids, to a cycle called the Calvin cycle. There we reduce carbon dioxide in a process where we fix the carbon dioxide to a molecule called phosphoglyceraldehyde. And you'll remember there's this sugar called RUBP and the enzyme called Rubisco. This cycle goes through and generates the sugars. These sugars are either stored in the leaves or then exported down into the roots. So remember, there are two sets of reactions, just like aerobic and anaerobic, but in this case there are two different reactions, one that requires light and one that requires carbon dioxide. In the light reactions, the important part here is that oxygen is split from water, and in the Calvin cycle you have the CO2 is being fixed or trapped into the sugars. So what we can do now is see how the working cells are all interconnected. So what we do is just kind of share with you this process. Let's start with the nucleus, nuclear membrane, and the endoplasmic reticulum. Here's the endomembrane system, along with this idea of DNA to RNA to protein. So in this case, you have a gene in a polar bear that forms for cytochrome oxidase. mRNA is going to transcribe that information, bring it into the endoplasmic reticulum, where it is translated into a polypeptide chain. That polypeptide chain then moves into the endoplasmic reticulum space and then to the Golgi apparatus. There, the Golgi apparatus can send that protein to the mitochondria. Or you can have a series of proteins that make enzymes to digest glucose. So in this case, glucose fructose kinase. And these enzymes can be made and stored in vesicles. We can then look at the cell parts or organelles like photosynthesis and mitochondria, where they cycle the carbon dioxide, the oxygen, the water, and the sugars. There they can be transported back into these vesicles where these digestive enzymes can digest the sugars and start that whole cycle over again. So at this point, what I'd like you to do is to visit the website about polar bears and treadmills and see how scientists are studying the oxygen use of the polar bear using treadmills and why that's important. At any point, come on back, visit the slides, check out what you need to do.